Welcome to G Fun Facts Online. The vast, silent expanse of outer space is famously hostile to life. It is a realm defined by a total vacuum, violent fluctuations in temperature, microgravity, and a relentless bombardment of cosmic and ultraviolet radiation. For decades, the prevailing assumption in astrobiology was that only the hardiest of microorganisms, such as highly specialized bacteria or the famously indestructible tardigrade, could endure such an unforgiving environment. Yet recent breakthroughs in astrobotany have shattered this microscopic ceiling. A humble ancient organism, known as the spreading earthmoss, or scientifically as Physcomitrium patens, has demonstrated an extraordinary ability to survive the vacuum of space. This fundamentally rewrites our understanding of extraterrestrial biology and paves the way for the future of off-Earth agriculture. When scientists from Hokkaido University and a consortium of Japanese research institutions strapped spores of this moss to the exterior of the International Space Station for 9 months, they expected sheer devastation. Instead, they witnessed a botanical miracle. Upon returning to Earth, an astonishing 86% of the unshielded spores not only survived, but successfully germinated and resumed normal growth. This revelation is more than just a testament to the resilience of terrestrial life. It is a critical stepping stone toward establishing sustainable human colonies on the Moon, Mars, and beyond. To understand how a fragile-looking moss can survive the lethal conditions of low Earth orbit, we have to dive deep into its evolutionary history, its cellular anatomy, and the cutting-edge science of astrobotany. Before we can appreciate its survival in the cosmos, we must first understand what makes Physcomitrium patens so unique right here on Earth. To the naked eye, it appears as a delicate, velvety green carpet, with individual plants reaching only a few millimeters to a centimeter in height, typically found in damp, open habitats across the northern hemisphere. However, beneath this unassuming exterior lies an evolutionary powerhouse. Approximately 500 million years ago, the ancestors of modern bryophytes, a group that includes mosses, liverworts, and hornworts, made one of the most significant leaps in the history of life on Earth. They transitioned from a comfortable aquatic environment to dry land. The primordial terrestrial landscape was a harsh, barren place, completely devoid of true soil and fully exposed to unfiltered ultraviolet sunlight and severe droughts. To survive, these early plants had to evolve extraordinary stress tolerance mechanisms. They learned to extract vital nutrients directly from bare rock, gradually breaking it down to create the very first soils, which subsequently paved the way for more complex vascular plants to thrive. Today, this moss serves as a premier model organism in plant biology. It is prized by researchers because its genome has been completely sequenced, allowing scientists to pinpoint exactly which genes are activated under specific stress conditions. Furthermore, it has a simple haploid-dominant life cycle. This means that, for most of its life, it possesses only a single set of chromosomes. If you think about human genetics, we have 2 sets of chromosomes, so a dominant trait can mask a recessive mutation. In this moss, because there is only one set, genetic mutations are immediately observable. Finally, it possesses an unusually high rate of homologous recombination, a sophisticated mechanism that allows researchers to easily perform targeted gene knockouts and insertions. Because of these traits, this moss has become the botanical equivalent of the laboratory mouse. But while laboratory mice are kept in comfortable, controlled environments, this little plant was destined for a much wilder ride. So why send moss to space in the first place? Astrobotany, the study of plants in space environments, is not merely a scientific curiosity. It is an absolute necessity for the future of human space exploration. As space agencies like NASA and private enterprises prepare for long-duration missions to the Moon and Mars, the logistical nightmare of resupplying astronauts from Earth becomes mathematically impossible. A sustainable, bioregenerative life support system is required. Plants are the ultimate multi-tool for space habitation. Through photosynthesis, they scrub toxic carbon dioxide from the cabin air and produce vital oxygen. They recycle wastewater through transpiration. They provide fresh, nutrient-dense food to supplement packaged space rations. Furthermore, numerous psychological studies have shown that the presence of green, growing things, significantly boosts astronaut morale, mitigating the intense isolation and sensory deprivation inherent in deep space travel. However, traditional crop plants like wheat, tomatoes, or potatoes are biologically expensive. They require vast amounts of water, specific light cycles, and nutrient-rich soil to thrive. Furthermore, they are highly sensitive to the stressors of space, particularly cosmic radiation and microgravity, which deeply confuse their root and shoot systems. Mosses represent a completely different approach. They are the quintessential pioneer species. They do not require deep soil. They can anchor themselves to bare rock, or even synthetic surfaces. They are incredibly efficient at utilizing minimal light and water. Most importantly, they possess an intrinsic, cellular-level resilience to extreme environments, a trait known as extremotolerance. If we are to one day terraform another world, or even just construct a self-sustaining greenhouse on the Martian surface, we will likely need to start the exact same way Earth did, with moss. But before sending the moss to the International Space Station, the Japanese research team, led by Dr. Tomomichi Fujita at Hokkaido University, had to select the right candidate. The life cycle of Physcomitrium Patens involves several distinct developmental stages, and not all of them are equally equipped for space travel. The researchers tested 3 specific biological structures against a gauntlet of simulated space conditions on Earth. They tested the protonemata, which is the juvenile, thread-like stage of the moss; the brood cells, which are specialized, thickened stem cells designed to weather difficult periods; and the sporophytes, the reproductive structures consisting of a microscopic capsule called the sporangium that encases numerous spores. In laboratory simulations involving deep freezing to -80 degrees Celsius, extreme heat of 55 degrees Celsius, high vacuum environments, and lethal doses of UVC radiation exceeding 100,000 joules per square meter, the results were definitive. The delicate protonemata perished almost immediately. The brood cells fared somewhat better, surviving freezing temperatures for about 30 days, but ultimately succumbed. The true champions were the sporophytes. Within the reddish-brown, rounded capsule of the sporangium, the dormant spores demonstrated an almost supernatural resilience. The researchers hypothesized that the complex, multi-layered structure of the sporangium acts as a biological spacesuit, absorbing the brunt of the deadly UV radiation and physically shielding the delicate genetic payload within the spores. Furthermore, the spores themselves are locked in a state of suspended animation, biologically primed to endure extreme environmental volatility until favorable conditions return. With their champion selected, the spores were integrated into Japan's Tampopo 4 mission in 2022. This mission was a broad astrobiological initiative designed to test the limits of biological endurance and the panspermia hypothesis. Dried sporophytes of the moss were meticulously fixed onto sterilized aluminum plates. These plates were then mounted on a specialized piece of hardware deployed on the extravehicular experimental platform of the Japanese experiment module, known as Kibo, on the ISS. Once the mechanical arms moved the payload into the vacuum, the moss spores were immediately subjected to an environment that instantly annihilates most terrestrial biology. Let us consider exactly what that environment entails. First, the vacuum of space. At low Earth orbit, the atmospheric pressure drops to practically zero. In a vacuum, water violently boils off and evaporates, instantly desiccating cells. For a human or a standard plant, this means explosive decompression and cellular rupture. Second, extreme temperature fluctuations. The ISS orbits the Earth roughly every 90 minutes. This means the exterior of the station is subjected to rapid, violent swings between the blistering heat of direct, unfiltered solar radiation and the profound, freezing cold of Earth's shadow. The moss experienced thermal cycling from roughly 120 degrees Celsius in direct sunlight to -150 degrees Celsius in the shade. Third, radiation. Without the protective blanket of Earth's ozone layer and thick atmosphere, the spores were bombarded by the full spectrum of the sun's output, including highly destructive UVA, UVB, and UVC rays. UVC is fiercely germicidal. It directly damages DNA by causing adjacent thymine bases to bond together, creating lesions that physically halt cellular replication. On top of that, background cosmic rays and high-energy particles constantly pass through, capable of shattering chromosomes. For roughly 270 days, 9 long months, the spores hung in the silent void, enduring a continuous barrage of multifaceted environmental trauma. The exposure plates were configured with different setups to isolate specific stressors, One set was fully exposed to the void and the sun, another was protected from UV light by a specialized filter, and a third was kept in complete darkness. Simultaneously, a control group was kept in a laboratory on Earth. When the exposure period concluded, the payload was retrieved and transported back to Hokkaido University. The researchers, well aware of the unprecedented synergy of space-induced stressors, braced themselves for a near-total loss. In a healthy, Earth-bound control sample, these moss spores boast a germination rate of approximately 97%. Dr. Fujita remarked that they expected almost zero survival. Instead, the results were staggering. The spores that had been kept in the dark or shielded from UV light while in the vacuum of space showed almost no reduction in viability. But most incredibly, the spores that had faced the absolute worst the cosmos had to offer, fully exposed to the vacuum, the freezing cold, and 9 months of unfiltered, tissue-shredding UVC radiation, yielded a germination rate of 86%. Under the microscope, the rehydrated spores swelled, their cellular machinery rebooted, and they pushed out healthy, vibrant green protonemata, as if they had merely woken up from a slightly longer than usual winter nap. The fundamental integrity of the genome and the viability of the organism remained gloriously intact. Based on these survival rates, the research team calculated that under these exact space conditions, the spores could potentially survive for up to 15 years. How is this possible? The survival of this moss is a masterclass in molecular engineering honed over 500 million years. The first line of defense is physical armor. The sporangium is heavily fortified with complex polyphenolic compounds that act as an ultra-high factor biological sunscreen. Much like melanin protects human skin, these compounds absorb the high-energy photons of UV radiation and dissipate them as harmless heat. But the greatest threat in a vacuum is the instant vaporization of water. Water is essential for the structure of proteins and the fluidity of cell membranes. When standard plant cells dry out, their internal structures collapse. This moss, however, is capable of a remarkable feat known as anhydrobiosis, which literally means life without water. When the organism detects severe environmental stress, often signaled by the rapid accumulation of the plant hormone abscisic acid, it undergoes a radical metabolic reprogramming. As water leaves the cell, the moss floods its internal cytoplasm with non-reducing sugars, primarily sucrose and trehalose. These sugars replace the water molecules, hydrogen bonding to proteins and lipid membranes to hold their three-dimensional shapes intact. As the dehydration becomes absolute, the sugary cytoplasm transitions from a liquid into a highly viscous glass-like state. This process, called vitrification, completely locks the cellular machinery in place, preventing cellular collapse and bringing all biological time to a standstill. Simultaneously, the moss synthesizes massive quantities of late embryogenesis-abundant proteins. These are intrinsically unstructured, water-loving proteins that act as molecular shields, coating the remaining vital proteins and preventing them from clumping together. Furthermore, in the presence of high radiation, oxygen and water molecules are split into free radicals, called reactive oxygen species, that violently strip electrons from DNA. The moss packs its cells with a robust antioxidant defense system, utilizing massive amounts of enzymes like superoxide dismutase and catalase to neutralize these damaging molecules. Finally, even with the vitrified cytoplasm locking the cell down, cosmic rays will inevitably score direct hits on the moss DNA, causing double-strand breaks. This moss boasts a hyper-efficient DNA repair mechanism, utilizing specialized proteins like RAD51 to locate broken ends and seamlessly stitch the genetic code back together the moment it is rehydrated. The successful survival of Physcomitrium patens outside the ISS has profound implications. When humanity establishes permanent bases on the Moon and Mars, we must learn to utilize the local extraterrestrial regolith. Lunar and Martian regolith are devoid of organic matter and contain toxic perchlorates. However, mosses are perfectly evolved to pioneer sterile, rocky environments. They secrete specialized organic acids that chemically weather rock, extracting essential minerals directly from the stone. As the moss grows and dies, its decomposing biomass becomes the very first layer of organic soil. In a temperature-regulated transparent dome on the Martian surface, genetically optimized strains could be cultivated to slowly break down the toxic regolith, sequester carbon, and generate oxygen. This moss-created substrate could then be seeded with complex crops, effectively bootstrapping an agricultural ecosystem from nothing but Martian dust and starlight. Beyond soil formation, moss offers incredible utility for the enclosed environment of a spacecraft. Integrated into the walls in sophisticated phagponic biopanels, it would serve as a highly efficient, passive, and virtually indestructible biological air scrubber. Should the spacecraft suffer a catastrophic power failure or a loss of life support, the moss would simply enter its dormant anhydrobiotic state, surviving the freeze and the vacuum, ready to revive the moment systems are restored. Perhaps the most mind-bending implication relates to the panspermia hypothesis, which suggests that life may have been transported to Earth from elsewhere in the universe on meteorites. For decades, critics argued that the journey through the vacuum of space and lethal radiation would sterilize any biological material. However, the revelation that a complex multicellular plant spore can endure the unshielded vacuum and UVC radiation of space for nearly a year, and theoretically up to 15 years, adds massive credibility to this theory. If a terrestrial moss can survive the void today, ancient microbial spores could have survived interplanetary transit billions of years ago. The journey of this humble moss, from the damp forest floors of Earth to the freezing, irradiated exterior of the International Space Station, represents a profound shift in our understanding of biology. For 500 million years, the intrinsic mechanisms that allowed it to conquer the barren rock of primordial Earth remained locked within its genome. Now those exact same evolutionary tools are poised to help humanity conquer the final frontier. As we look toward the Artemis missions and crewed missions to Mars, we will not be going alone. We will take the biosphere with us. And the vanguard of that extraterrestrial biosphere will likely not be a towering tree, but a tiny, indestructible patch of green moss, silently turning the dead rock of a new world into a home.